Hi there, and welcome to this video on GCSE Biology for the AQA specification focusing on cell division and in particular stem cells. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started.
Hello and welcome to tutorial 3 of 3 on cell division, and today we're going to be looking at stem cells. So in our last session we were having a look at mitosis and the cell cycle, and in that we covered interphase, mitosis and cytokinesis, and we also learned how the cell changes in each of these stages and in what instances mitosis is used, why it's so fundamental to the human body developing from a single fertilised egg. So in today's tutorial we're going to be having a look at stem cells, both embryonic stem cells and also adult bone marrow stem cells and plant stem cells, and we're going to summarise all of this by having a look at how we can use stem cells in terms of treatment, so in clinical practice. So first of all, let's have a look into the basic stem cell. So a stem cell is an undifferentiated cell of an organism which is capable of giving rise to many more cells of the same type, and from which certain other cells can arise from differentiation. So what does this actually mean? Well, first of all, let's just address the basic fact that a stem cell is an undifferentiated cell. So this means that it is not yet specialised. So it's just a blank slate, essentially, and therefore it has the ability to differentiate and form a specialised cell. So it can divide by mitosis to form a tissue of stem cells, and from there then different cell clusters in this tissue of stem cells can differentiate to many tissues of different specialised cells. So this is how the human body develops from a single embryonic stem cell, because remember we've got to produce so many different types of cells, whether that be lung cells that are specialised for gaseous exchange, or muscle cells that are specialised to contract. We've got to produce this from one single type of cell, and that's our stem cell. So let's move on to our second specification point of today. So let's have a look at embryonic stem cells. So stem cells in embryos are, funnily enough, known as your embryonic stem cells. And they're found in early embryos, and they too are completely undifferentiated. So what this means is that we can basically take this embryonic stem cell and turn it into any type of cell that we'd like to. So if we take a look at this stem cell over here, so it can undergo mitosis to produce many more stem cells, so basically increase its population, because remember last time we were looking at how mitosis forms two daughter cells from one parent cell. So in order to increase the population of stem cells, the parent stem cell can undergo mitosis to form many daughter cells. Or the stem cell can alternatively differentiate to form these specialised cells. So there's two options for the stem cell here. So just to give you a bit more context, so when we're talking about an embryonic stem cell, we're talking about a stem cell that comes from this stage of human development. So this is our embryonic stem cell. And so this occurs after the sperm and the egg have fused to form the fertilised egg, or in other words, the zygote. And from there, the zygote contracts and undergoes mitosis, and we get even more cells produced, and these cluster to form the embryo. And from there, this tissue of stem cells, as we were saying earlier, can then undergo differentiation and produce different tissues, different specialised tissues. So embryonic stem cells can be cloned and made into most types of cells. And environmental factors can be used to influence their differentiation into almost any type of cells. So evidently we can probably use this in clinical practice. If we can induce a stem cell to transform into any type of cell, or I should say differentiate into any type of cell, that could be really advantageous for us as clinicians. So just keep in mind here that you don't actually need to know any stem cell techniques. So if a textbook starts to lead you along that pathway, you can backtrack and just come right back to these tutorials. So now that we've had a look at embryonic stem cells, let's go and have a look at adult stem cells. So, next specification point. So adult stem cells are found in only a few places, so the bone marrow and the teeth, to name a few of these. So these cells cannot differentiate into any type of cell. So that's a crucial difference here. Embryonic stem cells, remember, can differentiate into any type of cell. But your adult bone marrow stem cells can't do this. Instead, they can only differentiate into a select few types of cells, especially blood cells. So your red blood cells, your platelets, which are responsible for clotting, and your white blood cells, which contribute to immunity. So what I want you to take away from that is that if you contrast adult bone marrow stem cells to embryonic stem cells, adult stem cells can only form a select few types of cell. So they can't differentiate into any type of cell, whereas embryonic stem cells have the potential to do this. So this could mean that perhaps embryonic stem cells may be more useful in a medical setting than adult bone marrow cells, for example, unless you are in need of blood cells. So now let's move on and take a look into plant stem cells. So stem cells in plants are found in the meristems, which is just a name for the tissue in plants that contains undifferentiated cells. So stem cells in plant meristems can differentiate to form any type of plant cell. So pretty much like the embryonic stem cells, we can form comparisons here. And importantly, they're present throughout the lifespan of the plant, which means that they not only give rise to various organs of a plant, but they are also responsible for growth. So now let's take a look into how treatment with stem cells may be able to help conditions. Oops, I'm getting a little bit ahead of myself here. So another important fact about plant stem cells is that they can actually be used to prevent extinction. So if there's any plants that are threatened by extinction, maybe due to global warming or due to deforestation occurring because of increased urbanisation in this growing world, then perhaps plant stem cells can be harvested and used to create these plants in a more sheltered setting and therefore prevent extinction of valuable species. So now let's have a look into how treatment with stem cells might be able to help various medical conditions. So patients who suffer from diabetes suffer from a lack of insulin. So this is particularly the case in type 1 diabetes. And this lack of insulin means that they can't utilise the glucose in their bloodstream. They can't uptake the glucose from the blood into their cells. And therefore they have this build-up of glucose in their blood, and this leads to high blood glucose levels. So stem cells can be made that can produce insulin in order to treat those who suffer from diabetes. And again, this is type 1 we're talking about. Type 2 is slightly different, but don't worry too much about that here. So this insulin that we are making from stem cells can be used in insulin injections. And if we inject this insulin into patients who suffer from type 1 diabetes, this means that they're able to now transport glucose from their blood into their cells, and glucose can now be used efficiently for cellular respiration. Whereas prior to this, without insulin, their cells were unable to uptake glucose. So in addition, stem cells can also be used to treat paralysis. So have a think about it. So someone is paralysed, perhaps their nerve cells are destroyed. So you can stimulate a stem cell to form a nerve cell. And these nerve cells can then be used to replace the damaged nerves in this patient and therefore contribute towards curing or at least partially alleviating whatever paralysis they have. So this is all some pretty novel techniques that we're talking about here. So they're not commonly used in clinical practice, apart from the insulin that we were talking about, but they are very much up and coming. So now let's have a look into therapeutic cloning, which is a way in which we can utilise stem cells. So a really, really big issue about medical treatment is the concept of immune rejection. So although the previously mentioned techniques for diabetes and paralysis are very effective, there is a risk that the patient's immune system will actually reject these cells because it senses them as foreign. So anytime a foreign body enters our cells, our immune system will detect it as foreign and therefore launch an immune attack against it. So think of your immune system as kind of like the police force of your body. So therefore, if we're injecting foreign cells, these stem cells, into a patient, the recipient, so the patient, might start to reject these cells because it identifies them as a threat to the body. So this is where therapeutic cloning comes in, because it reduces the risk of this immune rejection. So therapeutic cloning uses stem cells from the patient. So an embryo is produced to have the exact same genetic material as the patient, and stem cells are then taken from the embryo to treat the patient. So because the stem cell derives from the patient, it really should not be rejected by the immune system of the patient because it's detected as a self cell. So it's detected as a cell that has derived from the patient rather than as a cell that is foreign to the patient. So let's just have a look at this in a tiny little bit more detail. So as I said, with therapeutic cloning, we produce an embryo that has the same genetic material as the patient. Well, how do we do this? So remember, the genetic material is contained in the nucleus, which is here. So we're going to want to harvest some cells of the patient. We're going to want to remove the genetic information from the patient, remove its nucleus, and insert it into an empty egg cell, which we can see happening here. And then we're going to use an electric shock to stimulate the egg cell to divide to form an embryo. And so this cell here that you can see here will then undergo mitosis, rounds of mitosis, and form an embryo. And these resultant embryonic stem cells are then used in therapeutic cloning. And as you can see, they've utilised a nucleus from the patient and an empty egg cell from a random donor. So, now let's take a look into the ethical debates surrounding therapeutic cloning. So the use of stem cells has many risks, and people therefore have various objections to the use of stem cells in medical practice. So these can be pure scientific opinions, but it can also be ethical or religious opinions. So let's explore a few of these now. So stem cell research can be used to treat many diseases, including diabetes and nerve paralysis. So therefore, surely it should be utilised, because obviously diabetes and paralysis are very debilitating diseases. So this is an argument for using stem cells. Also, stem cell research can be used in research to aid discovery of treatments for diseases such as cancers, so we shouldn't be rejecting these stem cells that we can perhaps use in order to aid development of our current global medical landscape. Also, if we act to prevent stem cell research, it's probably just going to be driven underground anyway and might occur illegally, which is potentially dangerous if patients are starting to turn towards the black market, for example, to undergo stem cell therapeutics. So it's much better to legalise it and obviously heavily regulate it via various governing bodies. So now let's take a look at a few arguments against using stem cells as treatment. So, if we're using embryonic stem cells, we're essentially destructing what could be a new life. So, many religions believe that life starts after fertilisation. Therefore, if we're using embryonic stem cells, we're essentially killing a future human. So this is a big ethical debate that surrounds the use of stem cells. What gives us the right to kill people? And this is a common recurring theme throughout medicine. Also, if the sample of stem cells is contaminated by any kind of pathogen, such as a virus or bacteria, it could be transmitted to the patient, and obviously this would have negative side effects on the patient. And in addition, even if we do heavily regulate stem cell research, it may not be followed strictly inside labs. So these are a few points for and against using stem cells as treatment. I would suggest you go away and just have a little Google of it and a little read through articles about the ethical debate surrounding stem cells, because it is rather interesting, and this just provides a little taster of what is actually out there. But yes, so well done for today, and that's all.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.